the first part of a webinar series that's going to cover the endocrine pancreas, with specifically covering the hormones insulin and glucagon, and also discussing the condition of diabetes mellitus. So the first part, we're going to do a basic overview of just the pancreas. Now the pancreas is both an endocrine as well as an exocrine organ. The majority of the cells within the pancreas are composed up of these pancreatic acini cells, or acinar cells, which produce a number of enzymes involved in digestion. So when we cover GI physiology, we'll be talking about these particular cells. Also within the pancreas, you have some endocrine cells, and you'll notice here these are the islet cells, or the islets of Langerhans. Now, they contain different types of cells. They have alpha cells, beta cells, and delta cells. The alpha cells produce glucagon. The beta cells produce insulin. The delta cells produce somatostatin. Somatostatin you've heard of before. Somatostatin is also produced in the hypothalamus, and we looked at its role on controlling growth hormone secretion. But it's also produced within the pancreas, and it plays a role in mainly kind of helping to regulate the hormones such as insulin release. It is structurally identical to the somatostatin made in the hypothalamus. So what we're going to do first is look at insulin and its role in glucose metabolism and other I should say just metabolism in general. Sorry. Now, insulin is a very important anabolic hormone. It's not the only anabolic hormone we have. Anabolic hormones are hormones that promote building up, like growth hormone. Androgens, such as testosterone, is an anabolic hormone. Insulin is also an anabolic hormone. It is needed for normal growth. And when we covered growth hormone, we talked about how insulin, the need for insulin goes up when growth hormone is around. Insulin is a peptide hormone, so it's very important for this one you do realize it is a peptide hormone. Therefore, its receptor is located on the plasma membrane. So the insulin receptor is located on the plasma membrane of target cells. And in order for it to get the effects on the cell, we have to use signal transduction mechanisms to eventually get the overall effects. Now, the insulin receptor is an example of what we call a tyrosine kinase receptor. When the hormone binds a receptor, it has a tyrosine kinase associated with it that ends up phosphorylating these beta subunits, making this receptor active, and eventually through a series of steps result in changes within the cell. And the effects are widespread. One of the effects it does is it affects protein synthesis. It has effects on metabolism. It's going to influence the movement of transport proteins to the cell surface to allow more glucose to get into the cell, and we're going to go over those very shortly. But we're going to look at the synthesis of it first. Now, on the islet cells, and specifically the beta cells in the pancreas, the beta cells in the pancreas synthesize insulin. On those cells, they also have glucose transporters. Since glucose is a hydrophilic compound, in order to go across the cell membrane, you need some sort of transport protein. We have different variants of glucose transporters. On these cells, they're called GLUT2 transporters. Just for kind of keep that in mind for now, and we'll talk about the difference between that and other glucose transporters. These will allow glucose to get across the cell. The main stimulus for the secretion of eventually insulin from the cell is high levels of glucose. But you'll notice, according to this picture, there's some other factors that can affect insulin secretion. But when the cell receives its stimuli, eventually it's going to stimulate the increased production and secretion of insulin, and insulin is produced as a precursor hormone that through processing removes a few amino acids here and there. Eventually it assumes its proper three-dimensional shape and then will be released from the cell. Now, I'm going to go back here. When it's released from the cell, it will bind to its receptor and cause a number of effects. Now here, I just want to kind of illustrate the difference between the GLUT2 and the GLUT4 transporters. The GLUT2 transporters allow glucose to go across the pancreatic cells. If there's high enough blood levels of glucose, that will really stimulate more and more insulin to be produced. Now, when insulin reaches its target, it's going to be telling the target cell you need to take in glucose and utilize it. In order for glucose to get across the cell membrane in the target cells, you're going to have glucose transporters. Well, in, say, adipocytes or muscle cells, they have GLUT4 transporters. And one of the effects of insulin on those cells is to increase the production and translocation of these GLUT transporters to the cell surface, which is going to aid in glucose entering the cell. So if you've got a lot of glucose around, you want more of these transporters to transport glucose across the membrane. The GLUT4 transporters are sensitive to insulin, so you will produce more of those. You'll put more of them on the cell membrane in response to insulin. But GLUT2 are insulin insensitive because you don't just need it to move the glucose across, because this is the cell that makes insulin. You're not going to wait around for insulin to be made if that's the purpose of the cell. So there's differences in regulation of these glucose transporters. They're also different in their binding affinity for glucose, but that's more of a biochemistry issue, or for a biochemistry discussion. Now, the different things that can affect insulin secretion, even though, in bold, these are the main ones that influence insulin secretion. If you have elevated blood glucose levels, that drastically increases insulin secretion. Lower blood glucose levels will decrease it. So that's why I have it in bold. But there'll be some other things I do want to mention that influence the secretion of insulin. Now, if you look at this picture here, this is a picture taken from a biochemistry textbook that we used to use in biochemistry at this university. And I like the picture because it really illustrates a couple of major concepts. The note on the left, what they did is they administered straight glucose, a glucose meal, to an animal. They monitored blood glucose levels and the insulin levels. So you notice at the point where they took in the glucose, following that blood glucose went up, but eventually it started to go back down. Also, you'll notice the time they ingested the glucose, insulin secretion went up, and eventually it went down. So what was happening? Well, the reason why the blood glucose levels started to go back down is because you produced more insulin. And insulin's job is going to lower blood sugar levels. It's going to allow more glucose to get across membranes and for cells to use it. The stimulus for that insulin, and there's quite a big jump in insulin levels in the blood. The major stimulus is that glucose load. Now, if you look to the right, though, they had an animal that had straight protein. There were no carbs in the meal. Straight protein meal. Blood glucose didn't change. Well, you don't expect it. When you break down protein, you're going to get amino acids. That's not glucose. So glucose levels don't change in response to a protein meal. You'll notice there is a small increase in insulin secretion in response to protein meal. But if you compare this to over here, it's a lot smaller. So protein ingestion also increases insulin, but think about that's what you want. Insulin's job, besides affecting the glucose, its overall effects on growth. And we're going to be going through all the major effects, but think of insulin as an anabolic hormone. It's about building up. And in order to have growth, you need insulin. And so protein will also increase insulin, but there's a bigger difference between protein ingestion and glucose. You'll have a lot more insulin secreted in response to glucose than to protein. So if we go back over here at the chart, so we'll discuss some of the other things. Let's look at some of the GI hormones. The GI tract produces its own hormones. They're involved in controlling secretions, motility, digestion within the GI tract. So think about it. If a major stimulus for insulin secretion is being fed, you just got fed. I don't care what you ate, you were fed. You're going to have more insulin. Well, think about when food's going through the GI tract and you have increased secretion of these, it's saying, okay, looks like we're in the process of digestion. I'm going to probably need some more insulin to be able to deal with the end products of digestion. We mentioned about some of the amino acids when you have protein meal. Some are more potent than others. You need that for normal growth. Okay, if you look at these other hormones that affect insulin secretion, I want to emphasize these two here. Growth hormone, we mentioned in the lecture on growth hormone. When growth hormone's around, blood sugar levels tend to go up. So you need a little more insulin to be able to utilize that glucose. You also, when you think growth hormone, you think overall effects on growth. Insulin you need for growth, because when we look how it affects metabolism, how it affects muscles and things like that. So you kind of need them both together. Cortisol, we had talked about, is a major glucocorticoid. The glucocorticoids have effects on glucose metabolism. So cortisol tends to increase glucose levels. So if you have higher levels of glucose, that would tend to increase insulin a bit. Now the last one here is these particular drugs that are used in the treatment of diabetes, and specifically type 2 diabetes. What they do, their mechanism of action, without going too deep into detail, is they promote increased secretion of insulin by the islet cells in people with type 2 diabetes mellitus. Because with type 2 diabetes mellitus, it's mainly a problem with insulin sensitivity. If they require more insulin to have the same effects than in a normal person. So what they're doing with these drugs is trying to increase the production of insulin by the pancreatic cells in these patients. Now if you look at the things that decrease insulin secretion, it's a major, if you have a decrease in blood glucose, that decreases insulin. Somatostatin, it's like, that literally means stop growth. Well, if you don't want to grow anymore, let's have somatostatin around. This somatostatin is actually produced by delta cells that are found in the pancreas. Kind of keeps everybody in check. So what it's doing is it's keeping insulin in check. Fasting. When you're fasting, you haven't eaten, you aren't going to want to be building things up. If anything, you're going to probably want to be breaking things down. Your blood sugar levels may not be up there. We want to maintain blood sugar levels, keep them at an adequate level, so you don't want a lot of insulin around because that would decrease blood glucose too much. So fasting would decrease insulin. Now the last one is leptin. Leptin is a hormone produced by adipocytes, adipose tissue. It is a signal that does affect appetite. We'll talk about that when we talk about dietary balances at the end of the trimester. But what it also does is it decreases insulin secretion. It's a signal that says, hey, there's a lot of fat here. I don't need any more fat. So let's decrease insulin secretion, because one of the things that insulin does is it promotes fat synthesis. So this kind of keeps it in check. Leptin will also decrease your appetite. So this is kind of different things that can affect insulin secretion. And these are not the only things that affect insulin secretion. So you'll notice the stimuli for it include hormonal, it includes humoral, and the nervous system also does influence it. But the major stimuli for it is humoral. It's changes in blood glucose. Now let's kind of go over some of the major effects. So before you look at these and you look at the listing, think big picture. Insulin, I want you to think you've been fed. Let's build things up. Let's grow. Let's store things away for later. So overall, it has an anabolic effect on the body. So one of the places it's going to affect is muscle and fat cells. So one of the things it does that you saw in the earlier picture is it increases the production and insertion of GLUT4 transporters within striated muscle and adipocytes, which correspond to the majority of the tissue in your body, is muscle and fat. It will also affect the phosphorylation state of enzymes. They're involved in metabolic pathways. So it will be involved in either activating certain pathways or inactivating certain pathways. It also will promote DNA and protein synthesis. You think, okay, anabolic, yeah. So those things listed is, again, illustrated, let's go back, using this picture here. So insulin binding its receptor activates it, promotes protein and DNA synthesis, it has metabolic effects, and it increases the insertion of GLUT4 transporters into the membrane because you need glucose to move across, and then you're going to use it for various things. So let's look at its effects on muscle. Now muscle, when you're resting, you're not active, loves to use fatty acids. There's a preference for it. When you exercise, the utilization of glucose does increase. Now, when you exercise, the permeability to glucose increases, so you really don't need a lot of insulin when you're exercising. Just because of the movement, the permeability to glucose increases. At rest, the membrane permeability to glucose is just slightly permeable, so that's why it's going to use predominantly fat. Now, after you've eaten and you've got insulin around, one of the things it does is let's promote some glycogenesis. That means the formation of glycogen. So what it's going to do, it's going to think of it as carb loading. So you're going to have some glycogen around for when you do exercise. You're going to break down glycogen to glucose for the muscle to use. So it has its own supply of glucose. It's just storing it away as glycogen. Also, as I just mentioned earlier, during exercise, the glucose in your blood can go across the membrane relatively easily, but initially it's going to go and keep using predominantly the glucose created by the breakdown of glycogen. So after you've eaten, it promotes the storage of glucose as glycogen. One of the ways it does that is insulin, through its cell signaling mechanisms, is it's going to regulate the enzymes involved in glycogen metabolism. the rate-limiting enzyme in glycogen synthesis is active, the rate-limiting enzyme in glycogen breakdown would be inactive. And I'm not going to go any more in depth because it's something you would have covered in biochemistry. Also, following a meal, in response to insulin, you're going to favor protein synthesis. So obviously you're not going to have a straight carb meal. You're going to need some proteins in your meal because you're going to need amino acids to promote protein synthesis. But overall, you've eaten, insulin's there, it's definitely going to promote the formation of muscle, protein synthesis, and the storage of glucose as glycogen. So it's anabolic. In the liver, let's look at these bottom two here. In the liver, insulin favors the formation of glycogen because remember, the liver has a good store of glycogen for in case of conditions where your blood sugar levels get low, you can break down the glycogen for glucose to maintain blood sugar. So it's like you've eaten, let's store the glucose away as glycogen within the liver itself. Any excess glucose, or if you've had some fats in your meal and all that good stuff, it also insulin promotes the formation of fat. So it promotes lipogenesis. So in biochemistry, you learned about the pathway in which we make VLDL, and that goes out into the bloodstream, and eventually it finds its way to adipocytes, and we'll look at that in a moment. But it does promote the formation of glycogen, fat formation. It also, if you've got insulin around, you're thinking I got plenty of glucose. Well, the liver will utilize that glucose for energy. So you will have active glycolysis in the liver because if you got plenty of glucose, the liver say, I'll use it. And I use that energy in making glycogen, in making fat because they need energy for it. So in the liver, insulin favors glycolysis, also favors the formation of glycogen and lipids. That leaves us with adipocytes. Its effects on fat. Well, here's the VLDL that I talked about that the liver was packaging up. Remember, it's taking the triglycerides and packaging them up with some proteins and increasing their solubility and transport in the blood. It makes its way to fat tissue. And what it does, insulin, besides the chylomicrons, which are part of your dietary fats, they promote the fat synthesis within adipocytes. It favors certain enzymatic pathways and promotes the lipogenesis and the storage of fat. So overall, when you think insulin, you think I'm going to make some glycogen, I'm going to make some fat, I've got plenty of glucose, so if I can, I'll use it for energy. But overall, I'm storing things away for a rainy day. And also, we said it also promotes muscle synthesis or protein synthesis. So what happens? You haven't eaten in a while, okay? So we're talking about things that happen shortly after you've eaten. Insulin levels went up. Now it's been a few hours after you've eaten. Your insulin levels are going to start to go down. So you want to break down some glycogen because you need to maintain blood sugar levels. The liver is not going to be taking up as much glucose because its glucose is going to be used more, or there's a heavier dependence for glucose within the brain. Red blood cells can only use glucose. The liver can use something else. It can use fat. So certain tissues aren't going to be taking up glucose as much because it can use an alternative form of energy. It also, when between meals, when insulin levels are declining, you're going to favor the breakdown of fat because think again, you still always need energy. So you can get energy from the breakdown of fat. You still want to maintain blood sugar levels. So when you break down fat, you can use the energy from the breakdown of fat to provide the energy for the liver to to make glucose and gluconeogenesis. Okay, so those are different things that are going to happen between meals as insulin declines. So what happens if you're resistant to insulin? You have a receptor that's resistant to insulin, or you don't make insulin. So really, I'm talking about someone with either type 1 or type 2 diabetes mellitus. Type 1, it's insulin absence. Type 2, it's an insulin resistance. So kind of think about here, this is kind of listing mainly things in regards to fat metabolism, but we can kind of draw you a picture. You always need energy. The person who's not making insulin cannot utilize glucose effectively. You've eaten or it just can't, they're not making insulin, so they can't utilize glucose effectively. Some tissues have a heavier reliance on insulin than others. So your blood sugar levels are going to be up, but you still can't use it. So what are you going to use for energy? Well, it's break down fat. So you see a lot of lipolysis. These people can lose weight. Okay. You're going to use the energy from the breakdown of fat. The fatty acids, you're going to use beta oxidation to create energy. What also, though, when you're doing this, is you're going to have you can have excess of fatty acids being released as a result of elevated lipolysis. And that colipid and cholesterol production in the liver, which then are released as lipoproteins. Very high levels of plasma lipoproteins can increase the risk of atherosclerosis quite a bit. So they have an increased risk of atherosclerosis. In the process of excess breakdown of fats, the fatty acids are oxidized to acetyl-CoA. Acetyl-CoA in excess can be converted to ketone bodies. You have a lot of excess ketone bodies in your blood. That can lead to acidosis, so you can get ketoacidosis. So with insulin absence, you see all of this. This also could occur with insulin resistance. Again, if you're not able to utilize glucose effectively, you're going to need to use fats more for energy. But I'm not going to put the thing where if you've got insulin resistance, then you're going to lose weight. A lot of people with insulin resistance tend to be obese. And we'll talk about the difference between type 1 and type 2 diabetes a little bit later. Now this other one, I just picked this picture just to really illustrate glucose in the brain. And does the brain rely on insulin? The brain does not rely on insulin. The brain always wants glucose. The majority of your glucose needs go to the brain. So it's not going to be relying on insulin in order to bring the glucose into the brain and utilize it. Brain cells are very permeable to glucose. The brain always, even if you haven't been eaten 4, 5, 6, 7 days, huge requirement for glucose. It will use alternate means for energy, like ketone bodies, but it will not use fat. So after you're in the fasted state, starved state for a few days, eventually it can start using ketone bodies for energy, but still most of its energy requirements come from glucose. It's real important to have plenty of glucose around for the brain because the brain needs it to function. And just to illustrate this, these are PET scans where what these are monitoring, it's glucose. This is labeled glucose. Here's a normal individual. Here's an individual with mild cognitive impairment. Their usage of glucose is impaired. People with Alzheimer's disease, very limited ability to use glucose. And so this picture illustrates the brain has a huge requirement for glucose. Therefore, I'm not going to be dependent upon insulin. I'm just going to get the glucose. So the glucose transporters they have in the brain are not sensitive to insulin. They're just going to bring the glucose, and they have a very high affinity for glucose. So no matter if the blood glucose levels are getting a little low, they're going to try to get their glucose. So we're going to stop here for insulin. The next part is going to cover the effects of the hormone glucagon.